Hello, more raw content. So this is once again Skylar, and this video will mainly be focused on the large cell, or big cell, as the project says, that I showed off in the last video. Uh, don't mind background noise. It is a wonderful day outside, and everyone has their dogs. So we will do our best around that. So in this, we'll start by slow observing just for now, so I can watch. In the last video, so far, I've gotten a comment that was asking about the way that photosites in my project were splitting in unison. I'll be able to show that off pretty well here. Specifically that I am using a mod. This one is specifically called Enzyme, or this is a hotfix for Enzyme. It's I posted it on our Discord server. But Enzyme and almost every mod now of Cell Lab has a feature called minimum connections. In vanilla Cell Lab, there is maximum connections. So a cell will stop splitting if it has a certain number of connected or adhered neighbors. But there was no way without stem cells to easily check if a minimum amount has been reached, as in, don't split until I have this many neighbors connected to me. So in this example, while it continues to grow, I have the wrong setting on, so we will reload that and start watching again. We'll do regular observe, let it grow a little, and then freeze. So, in this organism, I have all of these photosites. They're just gray. Coloring looks a little weird just because of how Cell Lab does colors, and some cells have shaders on them by default that affect their colors, but they are, for my ease of use, colored gray in the genome editor. And these photosites are programmed to not split unless they have a minimum of 3 neighbors. So as we see, every single one of them has 2, right? One to the left, one to the right. That's why we put a little cap at the end. We have a little cap at the end here that is acting as the right side neighbor for this final photosite. This cap also has that same built-in setting, so it will split when it has 3 neighbors. So it's only this keratin here that has absolutely no... It just will never split. I believe I have it set right now. I'll have to double check that. And this side, though, is the continually growing side. So I have this blue cell here. Another thing that we have in a lot of mods is split until, or split this number of times. So this blue cell is set to 20. It'll split 20 times, and then at the end of that 20, it will change itself into a different mode, into a different part of the genome. Which is extremely useful. It's very limited because it can only do 20 splits, so I guess you could say 20 segments on an organism. Obviously, you could program any of these photosites to grow larger, to become a larger segment. That's how my Titan grows. It is made up of 20 segments, but each segment is quite large. But yeah, so we will see here. If we continue to slow observe, it should go to 20, so this might be a minute. We will continue to see this photosite grow. And when it reaches that 20, I believe this guy is set to 20, so we might have to observe. Just to go a little further. And we can also open up the genome, load. I do not have it named. I do. Big cell. F. We can see mode 1 splits to 15, 20. If we go to 15, it's all color coded. It's 17 and 19, and as you can see on the bottom, max connections 20, blah blah, whatever. and then it becomes 17. So 17 here should be it. There you go. Max number of splits is 20. Mode after splits, number 23. So yeah, this will split 20 times. We're almost there. I could fast forward it, but only having 2 speed settings makes it extremely difficult. So we're just gonna wait. uh, but I was speaking with massive mistakes on this that I would really like to challenge myself to do something similar to this without using a load of photosites and maybe in a nitrate lacking environment, which could be quite a fun challenge to make something not this large. This is quite large for such a concept. All right, there we go. So we see it did its final split. It changed into number 23, and 23 instead creates that keratin at the end. And then if we continue to slow observe, this guy should split sideways, giving this photosite 3, and as you can see, it all happens in like 1 tick apart. So if we freeze. So yeah, what happened was the blue photosite split in half, which gave this photosite 3 neighbors, so it split in half, which then gave the next one 3 neighbors, the next one 3 neighbors, and it just created this near instantaneous split. And at the end here, we also had the end split in half as well. So we get these 2 symmetrical caps. These are different modes, this end one and the back one. One is more of a reddish and one is a bluish color. So we do have a head and a tail. You could make it fully symmetrical, however you wish to do it. But this organism focuses mainly on creating a shell. So you'll see everything splits and puts a bunch of photosites in the cavity. These photosites will then split again to fill up the cavity even more, which we can freeze. While these inner detached photosites are splitting and filling up this cavity with mass. That's really all we're doing here is we've made this nice cavity, a fully enclosed cavity, and we are injecting a bunch of cells into it that are not connected to the main organism in any way. It might be interesting to see how this organism turns out, by the way, because we're on slow mode. Slow mode makes the math more accurate. This organism was designed with normal mode in mind, so we'll see what kind of shapes we get with the more symmetrical, accurate math. But we can see that the outer layer here has become keratin and more photocytes. To thicken up the outer layer. It helps prevent these photocytes from slipping into the gaps and escaping, in my experience. We'll see it split once again. There we go, and this is very important. So this is the most crucial part of the big cell. So the big cell uses RNG, random number generators. In this case, it's not random number generators, so I guess it's just randomly generated outcomes, so RGO, I guess you could say, or something like that. To accomplish this in Cell Lab, one of the methods of getting randomness, in quotation marks, is if you have 2 gametes, and we can actually do cell diagnostics. Oh, does it actually not tell you? Wow, diagnostics does not inform you that what the gamete's partner is. Anyway, that's fine. We don't need that. So the gamete, the red gamete, it matches with the green, right? Common sense. So they want to merge together. They do. But they're from the same genome, and they have the exact same mass as each other. So in the game has to make a decision. The red gamete has an outcome of, let's say, mode 30, and the green is mode 35 or something. These are not the actual modes they will become, but that is just random numbers. The game has to make a decision when these 2 gametes merge to prioritize the red gamete or the green gamete. I do not know the internal equation of CellLab of how it decides that randomly. It could literally roll a random number generator. I know that mass does play a role. If green has more mass than red, it becomes more of a 70-30% outcome. So 70% towards green, 30% towards red. And you can use that. I've done it before in sex-based organisms to decide gender, to have more males or more females than the other. But in this case, it is 50-50. So what we do is we have a 50-50, and we have a whole lot of it. You can see a whole lot of these. Not all of these will work. Sometimes this red one, like let's say these two, maybe if these two reds and greens, like right here, this red and green are right next to each other, there's a chance that they will merge together, and the opposite red and greens, so this red here and this green here, will no longer have their partner. So in the final organism, you will see a couple gametes that are just left behind from this process, which does partially aid in the randomness of this organism. So I am perfectly satisfied with how that works. And we can also see our outer membrane now has a layer of fat. I just did that for fun. This organism doesn't need the fat reservoirs because it is aligned in photocytes. Easily one of the most overpowered cells in the game. But this is for testing purposes, not for practical purposes. At the same time, the tail is developing, and the head is also developing into a sensory organ of sorts. It's purely cosmetic. This organism is cosmetic everything. Nowhere in the world of Cell Lab is this a practical organism. So if we watch the slow observe, we will watch as... I should explain that too. We'll see as the gametes start to try to merge together, their adhesions are pulling them together, but you'll also see there is half of them did not do this gamete cycle. Half of those photocytes stayed photocytes and are now turning into these stem cells, and then the other half are the gametes. So we purposely, right off the bat, have roughly 50% of the possible organelles that will spawn will become just empty space. They will not become organelles. Just to fill up the organism. We don't want it to be purely organelles. We want it to have some pseudo or fake cytoplasm of sorts or whatever flowing within it. So we have that as part of the 50-50. So we'll see these gametes do their merges together. Eventually, they take their time. As we see, those photocytes are still creating more. There we go. So some of the gametes merge and create their outcomes. And then as you see, these outcomes then proceed to split again into two more gametes. These are not the same red and green gametes. This is, in fact, a secondary stage. So what we're doing here is stage 1 is a 50-50. A or B is the outcome. And then A or B will do a secondary 50-50 stage. So that now gives you like a A, B, C, D outcome. So this allows, in a very small amount of genome space, some RNG to create 4 unique outcomes. In this case, there are 5, because again, we saw this happen, where some of them at the very beginning stage converted into cytoplasm. But overall, we have 4 organelles, so we need 2 stages. And you could do a third stage, which gives you 8. It's binary. So every stage you add, in theory, doubles the amount of possible outcomes that can happen. And give you some really unique options. After more of this gamete merging happens, we see the front develop into this muscle-filled keratin appendage that uses secreting cells to pulsate, just to make some cool ripple effects. And then the egg of the organism, this blue thing here, is generated at the tail. We can now start seeing that our gametes have made their rounds, And now organelles are beginning to generate. All of these organelles create glirocytes to merge with their neighbors, and then they connect. Now, before things get too crazy, we have a couple of the organelles already spawned in, so we'll look at them. It's a little hard to see, but you can see with our yellow organelle here, it is a Y-like shape. This is part of it. This is another one over here, still developing. And it has a secreting cell in the middle. We have a kind of W-shaped, or more like a U-shaped organelle here with 2 secretion cells, which is blue. And then up here is the red. A lot of people are interested in the red. The red is simply just a ring of stereocytes with a... It hasn't generated yet. It'll generate from this cell. But a ring of stereocytes that will have a secreting cell in the middle. So, oh, there it is. So this is a green one. The green one's just like an L or an I shape. And it has 2 fat cells on either side, and then the secreting in the middle. So each of these organelles will secrete signal 1, 2, 3, and 4. And all of these stem cells, all around the inside, will be looking for those signals in multiple stages. So some of the organelles will have more priority than others. Stage 1 will look for 1 or 2, signal 1 or 2. Stage 3 will look for 3 or 4. Or stage 2 will look for 3 and 4. I misspoke. Overall, that means that all of these stem cells inside of this cavity will respond or change their shape or their outcome based on what organelle is nearest to them. And that kind of shapes the organelles. And so if we not do that, we continue to observe, we'll watch the red organelle put its secretion cell in there in a second. It's taking a while. There we go. So it's a glirocyte that then glues itself and then turns into the stem cell. So we'll continue to watch in slow motion, just because it's very interesting to watch. We should see all of the cytoplasm stem cells turn into glirocytes at any moment. And then all combine together. So we're seeing all of these randomly generated organelles forming. And there's quite a lot of them in this one, so they're very tightly packed. So we get a better look at the green, the yellows, the blues, and the reds. There's 2 reds here. There's a red over here, a couple of reds here. Oh, and now we're starting to see it. So now all the cytoplasm is starting to turn into glirocytes so that they can merge together. That will allow the signals of the organelles to travel throughout the organism. There you go. So all of our cytoplasm multiplies purely to fill the space even more. This will cause the organism to kind of swell in size now that all the organelles have, situated themselves. And we got quite a good distribution of the different ones. So you can watch as the cytoplasm ripples through all of its stages. You'll see this kind of ripple effect occur. Which that was one of the ripples. And at this point, I think we'll start to see these stem cells make decisions based off of what organelles are in proximity to them. This does mean that some organelles will fight the proximity. Like, I think red goes first, so this upper area is going to get a lot of red organelle concentration, but none of the green and yellow and blue. But we will see. But yeah, so far, if you've noticed, there's nothing really complicated happening here. It's just a giant cavity that gets created, full of stem cells, and then just they all connect to each other and just wait for input. The hardest part of this organism is just the randomly generated organelles, and even then, it's just some gametes that are fusing together. It's honestly not too difficult. I would consider this quite a bit simpler than the hormone gradients that we kind of talked about. This kind of does that. Each of these organelles does create a gradient around them, but it's not. It's random. We don't have control of this. We are allowing it to create itself, pretty much. So honestly, creating this only took me, I think, about 4 hours to make this organism. While in the previous video, uh, that smaller one with the gradient, took about the same amount of time, despite being significantly smaller in size. All right. So we saw some decisions get made. We're starting to see these stem cells turn into, these are flagella, these pinker cells, so that the red organelles can give those flagella signals to swim using those stereocytes. So all of these red organelles are designed to create flagella or convert nearby stem cells into flagella, and slowly steer the organism towards food. This organism doesn't actually need that food, but it's for cool factor. As we can see in slow-mo, actually, the front appendage broke, which is interesting because I've not actually seen it do that in normal mode. So slow mode is obviously affecting how this outcome occurs. So we're kind of watching this together. I don't know what this one will look like. Every single one that you spawn in looks a bit different. If you want to get some pattern recognition, we can look. So in this example, there's a lot of red organelles up top, a lot of green and yellow and blue fighting in the middle, and then red kind of in this bottom left. So if we want to memorize that. If you've noticed by now, the organism has swelled to quite a size in the petri dish. I believe this is a 2 mm dish. I could be wrong. Maybe 1.5. All right, phase 2. So around the yellow organelles, we are generating keratin. Around the green ones, we are generating photocytes. And in the final phase, the blue, the stem cells near the blue should start forming muscles. Which is interesting. So we got a lot of keratin in this organism. So make note of that too when we spawn a second one, that this one has a nice stripe running down the middle of keratin. So the keratin is set to have strong adhesion and such. It helps hold this organism together. It's less squishy, as a higher, it has a stronger cell wall in the settings. So the more keratin that generates, the stiffer this whole organism becomes, which will affect how it flexes and moves as the flagella try to move it towards food. So as these generate, you see unique, not behaviors, but like turning radius and stuff like that. We're starting to see the muscles form. These blue organs are constantly having the secretion cells alternate between different signals, so you will see these muscles kind of contracting and convulsing. Purely for visual, that it doesn't actually help the organism do anything. Once again, this is not practical. This is purely for cosmetics, and we'll see that it got so bloated in the back that it actually damaged itself and became disconnected partially from the organism. I believe everything does still have keratin on it, so unless an organism slips between here and starts eating all this, it's still a protected organism. The final stage of our fake cytoplasm is it starts to turn into white buoyancy cells. I only do this because the buoyancy cells kind of look like vacuum bubbles. You know, they're little bubbles of air. They're mostly transparent too, so depending on the light level, you know, change. You can see through the buoyancy cell, so it kind of looks like liquid, even though it's not. I do wish Cell Lab just allowed you to like fill a space with like a dummy cell that is just a liquid that like doesn't necessarily die of cell age. Maybe that would be kind of interesting, just so you can fill a cavity with fluid made by an organism, like actual cytoplasm. But yeah, so now if we observe, it's pretty much done. Now we can see the overall function of this organism. So we have all the movement from our pulsating blue muscles, and then we have all of our very squishy cytoplasm, and not all of it bound together, which is actually good because it creates some flexible spots. We have plenty of keratin, and then we have a little bit of flagella. This one didn't generate a lot of flagella, so it's not gonna really move much. But yeah, we'll watch that entire thing in observational mode. So remember this shape. Lots of keratin up front, down the middle, a whole lot of yellow organelles, and then we have a lot of red as well. And now if we reload the plate, now the math is different too, so this is a pretty bad example, technically. We can see just how fast that whole process actually happened. Slow mode really does slow things down. So immediately, as you saw, it takes literally 2 to 3 seconds for all the organelles to be made. So now we have a big U shape of red in the middle and a lot less of the yellow than before. And we have a lot more of the blue and green. So now we watch it as all of it grows. And there we go. So it does not take long for this organism to grow, and we got a whole new random one. This one has way more flagella in it too. And as you saw, the front did not break like it did last time in slow mode. This back did still break. It's very odd because I didn't have that problem when I designed it. So maybe I saved an older version that has some bugs in it. I'm not aware. Things happen. We'll observe another one just to see if it looks any different, which in theory it should. So we do some RNG. This one's way more even in its dispersion, as you see. Like, that's a nice one. I like this one. I especially like that there is a red organelle right in the middle. So that's pretty cool. This one also, I think, got less muscles. And as we see, the back didn't break at least as early in this one. And the reproduction of this organism, it's very superficial, very fake. It's literally just this egg is on a timer. That is roughly mathed out to the death of all these cells. And when that timer runs out, it splits into 2 M1s and just creates 2 of these big boys. So, by no means is it a realistic reproduction, by any means. And if we do look at the blue, we can see that there are stem cells on the blue organelle that change color. I don't know why this one didn't, but you'll see some yellows. So yeah, there's a lot of yellow stem cells. So I do have these stem cells on the blue organelles, keeping track of the age of the organism. So it goes blue, then yellow, then orange, and then red. And red, as you can assume, means it's very close to dying of old age. So if we actually incubate, you'll see that orange appear eventually. It's not going to run very fast even in incubate. And you can see we're kind of spinning a little bit from the muscle gyration. So we're in orange territory. And it's also spinning because of the flagella as well, which is nice. And you can see there's a couple gametes still in there that never fused, but that's fine. And now, there we go. Now we're in the red zone. It splits to make babies, which obviously this is not a big enough dish to do that, so they're all both deformed, and then the original dies. They do look very fascinating, though, when they are just exploded open. It's just a dead cell with all of its guts out. That's pretty hard to create naturally in this game. So, yeah, this is a much more chaotic video of me trying to, without going too in depth of the genome, like opening the genome editor and doing the most grueling, slow tutorial ever. I just wanted to show off the basic concepts of how this organism randomly generates, because when I posted this throughout Discord, quite a lot of players were interested in a video like this of uh, kind of how it works and possibly why I created it, or if there are any tips or tricks in here. Not necessarily. I mean, I think the biggest trick that was used in this is that very initial growth of using the minimum connections to form a chamber. You can also kind of use that when making like capillaries and arteries and stuff like that, for blood to travel through. You can use it on a small scale. You do not need to use it on a large scale. Um, let's not save that. A good example of that. We'll show another one. I showed this in the last video, I believe. But we'll show it again. This exact same method can be seen in my directional algae. So you got that free floating from the last one. But I did have unplanned video, right? I did have another one. There it is. This I've not made a video on. But this is a smaller scale organism that does the same thing. So it's two chambers, left and right, with a nervous system running down the middle that does that exact same process of gluing everything together and then generating a bunch of stem cells that then decide what to do. The only difference on this organism is that the outer wall has secretion cells making S1 and S2 so that the organism can differentiate the left versus right, so that it can like avoid walls and such. This was kind of mimicked after the real life algae called Phacus. It is an algae, a kind of predatory almost algae with a big red eye spot in the front and this very like disc shaped body with a tail. This guy's not going to avoid the wall very well in time. But see, these methods can be used to make intelligent organisms, so he will attempt to swim away from the wall before death. Just not very good at it. It moves very slow. But yeah, so this video is made to show off these general concepts. I hope that you enjoyed, or yeah, there you go. I hope that you enjoyed this video in particular. I'm going to keep doing this unedited raw. It's very easy for me to do. It's very relaxed for me to do. I don't have to plan, lazy, ADHD, all that fun stuff. But tonight, I think I might try to cook up a smaller scale organism that continues to use this chambered concept in a more practical way, more on the scale of that Phacus that I created, but maybe just visually different looking without photo site spamming. is hopefully the goal I'll reach eventually. But yeah, I hope you enjoyed this video. Hopefully you learned something from it too, especially. And that this was easy to watch. So I appreciate you coming. Have a good night.